Hello everyone, I'm Anisha Kirkia, Product Manager at NanoString Technologies. Today we have with us Matthew Gutierrez, who is a Principal Scientist at Mitobridge, a biotech company in Cambridge, US, that works on small molecule therapies to enhance mitochondrial function. Matthew's team led the biology effort on a novel therapy for Duchenne muscular dystrophy, a program now in phase 1. His research background is in muscle biology and cardiac development, and he received his PhD from Duke University. Today, he is going to discuss about the challenges with gene expression profiling in biotech setting and recent technical advances with NanoString PlexSet technology. So without any further delays, I will hand over the webinar to Matthew.
Thanks, Anisha. 5 years ago, if you would have asked me what the biggest challenges were with qPCR, I probably would say reproducibility. So this has always been something that has bothered me about the technology, and it basically required an increase in the number of technical replicates in any given experiment in order to feel a little bit more confident that the data you're getting are representative of the biology. Coming into biotech and working at a company that has a pretty tight focus on gene expression analysis, qPCR was really on my mind. And I knew about NanoString technology, but at the time, NanoStrings was able to run smaller sets of data. And so it wasn't something we considered when starting the company. We really focused on a very small picoliter scale qPCR system. The presentation today that I'm going to tell you about was essentially our decision-making process around purchasing a NanoString system when PlexSet became available, and some of the data that we generated while QCing that system. Disclosure, I am an employee of Mitobridge. Mitobridge is a biotech company located in Cambridge that focuses on small molecules to improve mitochondrial function. We're looking at multiple targets in and around the mitochondria with the goal to take these small molecules and put them to use in rare diseases and some not so rare diseases. We started just a few years ago, and we've got a small team. So as I mentioned, one of our key needs in setting up the lab was to be able to analyze gene expression. And we were thinking about some of the things that really were of most interest to us and our key needs. So that's matched here. So in thinking about the cost of each experiment, and that's on everybody's mind, it's not just the per sample or per endpoint cost. So how much does it take to run a given sample in your experiment? But it's also how you can take the results from any given data point and compare it to results that you got on a different run on a different day. And so a lot of times in my experience with qPCR is you really can only make good comparisons within an experiment or within a run. And so in some ways this requires taking the same sample and running it multiple times, and that of course will increase your cost. Sensitivity is a key issue. As we work in mitochondria, a lot of our targets are related to metabolism. And if you hit the right pathways and enough genes within it, really small changes in gene expression can have big changes on cell biology. And so here I've mentioned that changes as small as 20% should be measurable. And in some cases, it could be even smaller and have real effect on biology. And in my experience with qPCR, it's really larger changes that are those that you can see. So twofold changes and greater. So that didn't quite cut it for some of the pathways we were interested in. Next, throughput. I mentioned earlier that the NanoString technology at one point was able to test samples in a smaller set, so around 12 samples per run. But a lot of our experiments are either done in cells in a 48-well or a 96-well format, or in an animal study that can include many, many animals, and I'll give an example of that in the next slide. So throughput's really important, as well as flexibility. We run experiments in rodents, non-human primates, and human samples. So we need to be able to transit between these matrices and to be able to look at multiple pathways and disease panels. And finally, in terms of flexibility, the assay should be something that we can take from our preclinical setting and move into the clinic, a method that is already okay to use in a clinical setting. But foremost, and I think this is a concern any scientist has, it's reproducibility. And it's overcoming these hurdles that we experience with qPCR. And those are technical. How many replicates do we need to spend space on in a given experiment to make sure that we're getting the right results for a given sample? Biological, can we see a drug effect in the same way across samples? And experimental, can our experiments be reproduced on a day-to-day basis or by labs elsewhere? So here, this is a description of a typical animal study that we're running. It's one of the larger ones, but it gives you an idea of why this can be challenging in a qPCR system if it is not able to handle a large number of samples. So in this experiment, we have multiple groups that are challenged with or tested with either a compound at different concentrations, low, medium, or high, and a vehicle group. And we also have 5 different time points where the animals were taken down at different time points post their dose. And so with an N of 4 per group and per time point, this resulted in 110 animals. From this experiment, we had a baseline tissue, in this case blood, terminal blood, and a tissue sample that we analyzed. So that took our 110 animals and created 330 samples. And if we were to test 24 genes, including some reference genes, this required many runs, even on our picoliter scale system. So this increased the amount of time it would take us for us to get the results from this big study. And it also creates a challenge for the scientists because you have to start making decisions as to which samples need to be included in a given run. As I mentioned before, because of technical reproducibility being low and experimental reproducibility being low, you really need to make some conscious decisions as to which comparisons are most important to make. And so here's some data to back up the reasons why we are concerned. This is looking at our previous system, which is a standard qPCR. This is really no different than any other qPCR system that you might run into, with the exception that it's run at a picoliter scale. And these are 2 different experiments that are looking at 2 different types of reproducibility. So on the left is looking at technical replicates. Now, most qPCR is run in a way where you have a given sample run multiple times within the experiment. That might be 2 or 3, and the average result is usually recorded as the result for that sample. And that's because there is an appreciation for a small bit of noise that's within the experiment itself. So here you can see there is correlation. We have a significant correlation between the technical replicates. However, the R-squared value is a little less than to be desired, so it's 0.65 in this case. And in this case, we were looking at about 20 genes being profiled. Now, the experiment on the right is one that's, I think, a little bit more interesting. So 2 scientists within our team, both of which have plenty of experience running qPCR experiments, were given the same sample. So this was a blood sample that was split into 2. And both scientists independently isolated RNA, generated cDNA, and ran the same panel of genes on our qPCR system. And then we took the results, and in this case, represented in fold change for each one of those samples, and tried to match it up. And this is really where the big concern came in. You can see that the results don't correlate at all between the 2 different scientists, which means that the person who prepped those samples made a bigger difference than the biology that was at play within the experiment. And so in thinking about any experiment that you want to be able to be reproduced by other groups, this is a big concern. This is the exact same sample, and this is done by 2 accomplished scientists using the same equipment within the lab. And so this was one of the big challenges that we were facing before we introduced the NanoString into the lab. The next challenge was sensitivity, and I mentioned this before in one of the earlier slides, is that we need to be able to reliably detect small changes in mRNA. So here's an example of this. This is an experiment in cells. These are patient-derived fibroblasts, where we're looking for an upregulation of a key metabolic enzyme. And the compound that we're using here directly increases the transcription of that gene, and it should result in an increase in the protein, and we saw that by Western blot. So the Western blot results are looking at our protein of interest in reference to GAP-TH, and we saw a consistent upregulation. This is one example experiment across many experiments. Densitometry results from several experiments are shown in the graph on the bottom, showing at 3 and 30 nanomolar of our compound, significant increase over the DMSO control. But this increase in the amount of protein could not be detected when looking at the qPCR, which didn't make a ton of sense. This is a transcript that should be resulting this is the transcript that should be resulting in the increase in protein that we're seeing. But our methods just weren't sensitive enough to pick this up. And I think anybody who's done enough qPCR will recognize that this variability is pretty normal. And we usually attribute it to it being just normal stochastic responses within cells that are just slightly different. It's hard to pick that up. However, that never really sat well with me because these are the same cells, just separated by a different well. So that was a really a head scratcher, and it's something that you kind of lived with and hoped that the results would reproduce. So after trying out the NanoString system, and we worked with the Proof of Principle lab on some experiments early last year, the NanoString was purchased and added to the lab in May 2017. And really the decision maker for why Mitobridge added a NanoString was the advent of the PlexSet. And so no longer is really a lab restricted to the number of samples. The PlexSet allows you to run really any number of samples up to 96 for as many genes, up to 96 genes at once. And this can create a really large data set, and all of it with the NanoString technology that we found is much more technically reproducible as well experimentally. So when the machine showed up in the lab, I was very excited, and the lab decorated it. That's the reason for the balloons here. So in the first 6 months of use, we ran this machine pretty much nonstop and developed multiple PlexSets. So all of our previous primer sets had to be ported over to a NanoString PlexSet system. This was really a painless experience. Essentially, we generated short lists of primers up to, in this case, 24%. We can do more at this point, but this served the purposes at the time. We've looked at human, mouse, and rat PlexSets. These sets are sent over to NanoString's bioinformatics. Within a day or two, we get results back from them, which is essentially a primer list that you can submit for synthesis. And the generation of these PlexSets was fairly easy. So we've looked at over 250 genes with the NanoString in many, many experiments. And I'll show you some examples of that in the next few slides. The workflow for building these PlexSets are, I mentioned a moment ago, you design the sets of genes that you want to look at, and you should have about 4 or 5 reference genes included. Those are sent to NanoString's bioinformatics team. Probe set generation, basically oligos, is sent to you in the form of an order form, which you can send to any oligosynthesis company. We use IDT. For any new system, so a different tissue or cell type, we generally run an RNA titration experiment to give an understanding of how much signal we're going to get. And this is really only important if you have some genes on your set that are very highly expressed. You don't want saturation to occur. And then you're off to the racetracks with PlexSet experiments. So the experiments that I was describing earlier, we reproduced with the NanoString PlexSet. And so these are the exact same samples, the same RNA samples that were now run on the NanoString platform. In this case, we took those same samples and we profiled about 19 genes on the NanoString and looked at technical replication. And like the qPCR system, we do get nice correlation. And in this case, the R-squared value is much improved of 0.97. And the real deciding factor in making us happy that we incorporated the PlexSet into the lab came from the experimental replication, biological replication experiment. So I'll remind you, these are the same samples that I described before, tested by two different scientists. So the same sample split and prepped for RNA. And we took those ones and ran them on the NanoString. And you can see that the correlation is quite good. It's nearly perfect. So our R-squared value is 0.96 again. So what this means is that the lack of correlation that we were seeing previously had something to do with steps in between the RNA isolation and the actual data that we were getting in our previous qPCR system. What this data tells us is that the RNA itself can be prepped by totally independent people, and you'll get the same result. So it confirms that we're able to get good experimental replication independent of the person running the experiment, which is ideal for any science. In terms of throughput, the PlexSet Capacity really does meet that need. However, I mentioned before the need for running many technical replicates within a qPCR system. So if we needed to run, say, 3 technical replicates, those 96 samples would quickly get cut into a third if you had to run a technical triplicate. To be sure that that wasn't the case, a sample set was run in technical replicate, and in this case it was run over 3 consecutive days. And we looked at the correlation for the same samples across all 3 days. And you can see here that the data correlates quite nicely, the technical replicates, and it's replicating quite well across runs as well. So our R-squared value is essentially 1. And this type of data convinced the lab at Mitobridge that we no longer needed to run samples in technical replicates. We could cut that number from over 3 down to 1. Occasionally, if you get an odd result with an RNA sample, usually what we'll do is pull that sample, recheck the RNA integrity, and then go ahead and rerun it in the next step. But that actually doesn't happen very often. So by cutting down the need to perform technical replicates, the throughput's actually quite a bit higher, and we're able to get much more data per experimental run. This really does cut down our costs. Then getting back to this experiment that I described before, and this is with fibroblasts in a dish. And you can see that the PlexSet data is able to detect the difference in the RNA, and we demonstrated an 84% increase in this target gene. And what's really nice to see is that the variability that we were picking up previously in these same samples turned out to be all technical in origin, and that the biological replication across these 4 independent wells of the same cells treated in the same way is almost 0 looking between these 2 groups. And this makes a lot of sense. They're the same cells treated in the same way. The genes should be going in the same direction. So this was a comforting data set to see, and we've been able to detect much smaller changes with the PlexSet since then. In terms of throughput, those large experiments I was describing before are no problem, and so this is just an example where you can take a large number of experimental groups and run them on the PlexSet and be able to compile the data across runs. So in this case, the experiment was run across 3 independent runs and all looked at together using the NanoString data analysis platform. And we're able to create one data set from over 330 samples looking at 24 genes at a time. So in summary, we've had, at this point now, a little over a half a year of experience running the NanoString PlexSet and many, many experiments under our belt. And this came after a successful set of experiments at the NanoString Principal Lab. And the conclusions that I shared with you are in comparison to our previous qPCR system that was a high-throughput picoliter scale qPCR. Overall, we've seen much improved sensitivity using the PlexSet. We can detect changes that we really couldn't detect before, all the way down to 15%. And this is due in large part to a reduction in technical variability. The throughput's improved quite a bit. Not only can we increase the number of samples per run, but because of the increased experimental or decreasing experimental variability, we're able to much more easily compare results across different runs. And so this means that the same sample doesn't need to be rerun in order to make new comparisons. We can make comparisons across runs in a much more straightforward setting. Overall, the flexibility has met our needs. We are able to port many different types of assays and different types of for different types of gene sets, different species, without any problem. And we found that adaptation of particular gene panels or PlexSets are fairly straightforward. An example of this is when a particular reference gene, which worked really well for PlexSet in one tissue type, is no longer as good in another. You might see that gene is changing in a way that just makes it inappropriate to use as a reference gene. Swapping out those poor reference genes for a new one that works is very simple and can really get a lot more distance out of any given PlexSet. And it allows you not to have to compromise on the normalization genes that you're using. And finally, it's really the reproducibility that I've been most impressed with in using the PlexSet system. So the inter and intra-user variability is minimal. I feel much more comfortable that a result that we see on any given day is not only going to be reproduced by us within the lab on a later time point, but also will be reproduced by others that use our compounds. And it was really enlightening to see that a lot of the variability that we picked up previously in qPCR, and something that in the over a decade of qPCR experience that I have, that I've always seen and attributed to biological variability, turned out to be mostly technical. And I think that this is something that anybody doing a lot of gene expression should consider. So with that, I'm going to finish and take any questions.
Perfect. Thank you so much, Matthew, for discussing the comprehensive view of the Encounter technology, advancing your research. So I do have a few questions over here. The first one is, how many genes did you measure per run?
At this point, we've been running about 24 genes per run, and when we first started working, the gene sets were 12 or 24, but more recently the PlexSet's been expanded so that we could run up to 96 genes per run. And this creates a lot of data, and it's, I think, going to be useful in some settings, but at this point, mostly 24 genes per run.
Perfect, thank you. And did you had to optimize these gene target probes before using with the PlexSet platform?
There's Hasn't been a need to optimize any of the genes. The only experiment that we'll do is an RNA titration experiment to make sure that we're not saturating the probe. But in terms of optimization, something that we used to have to do a lot of with normal qPCR, there is no need for this particular system.
Perfect. Another one, how do you manage or organize all the probe sets that you're currently using? Because you also mentioned about swapping few and also doing multiple experiments. So how do you really manage and organize these?
Yeah, this was a trickier thing, in fact. The lab, once the technology was demonstrated, was all on board, and everybody wanted to start running their experiments on it. And this made us both happy to get the system, but also a little worried about all of the PlexSets that would be floating around the lab. And so my recommendation to any lab that gets one of these systems is to have a plan in place well in advance of the system arriving to your doorstep of how you're going to manage all of these PlexSets that are generated and where they're going to live within the freezers so that there's no confusion as to which PlexSet is which. And so we generated a fairly simple Excel database that everyone has access to that we keep track of all these samples. But for a lab that's running a lot of qPCR, it's definitely something that you want to consider before starting the experiments.
Perfect, thank you. And how long did it take you to analyze your results? Let's say you talked about an experiment for your 330 samples. How long did it take to analyze using the Insolver software from NanoString?
For those larger experiments, it takes a little bit longer. It can take a few hours to analyze and feel comfortable that all the normalization is acting the same across the board. And so it takes a part of an afternoon. It takes much longer to start graphing out all of these results and interpreting what they mean. The Insolver software is pretty quick and easy to use.
Perfect. And then also, once you extracted your RNA, do you have to convert that into cDNA in order to put this into the PlexSet workflow?
Nope. In this case, we're directly measuring RNA. And the experiment that I was describing with the two different scientists probing the same sample, that is of the RNA directly. And the qPCR results was that same RNA made into cDNA. And so the NanoString system can directly measure RNA, and I think a lot of the variability in qPCR comes from cDNA generation. Very small differences from scientist to scientist in how they set up their cDNA reaction, I think, can make a big difference in how your genes are detected later on. And so that's, I think, a big benefit of directly measuring RNA in the NanoString system.
Thank you. This was useful. And one last question that I have over here. What system are you using? I know NanoString has a couple of different instruments. So which particular system are you using for your research?
We're using the Sprint. And I think that this is one of the newer systems. I'm not sure on it. Yeah. Yes.
Great. Perfect. Yes. So it's Encounter Sprint Profiler. Thank you so much. Yes, and these are it for the questions. Thank you so much for your time and providing such insightful information to the audience. I'm sure it will be very helpful and useful for them. So thank you once again.
You're welcome.